We all know genetics, so our sequence of DNA that gives rise to our different genes. And epigenetics is really what I like to think of as the operating system of the cell. It's what gives each cell its different kind of defining characteristics and phenotype. So even though the cells in your skin and the cells in your brain have essentially the exact same DNA, what makes them different is the epigenome. It gives them their overall function and structure. One form of epigenetics is called DNA methylation. Scientists found that DNA methylation is changed quite dramatically with aging. And using things like machine learning and AI, we've actually been able to predict how old someone appears based on DNA methylation. And this has been referred to as the epigenetic clock, which is basically just a way to try and quantify biological age based on either gains or losses in methylation at specific regions throughout the genome. We always think of the movie Benjamin Button, where he aged in reverse. I don't know if scientifically that is possible, especially at a whole body, whole organismal level, although we do know that you can reverse the age of a cell. Every cell in our body has a very specific function, and this function is really dictated by the epigenome. The problem is that with aging, the epigenome becomes remodeled, either due to stress or random errors. And what this produces is that each cell is actually going to lose its identity and not function in the way it was initially intended. And over time, as more and more cells become dysfunctional, you can imagine how this would produce dysfunction at the organ level and eventually at the whole system level. And the epigenome is highly dynamic. These are things that can go, we think, in both directions. So you can increase epigenetic age, but we've also shown that you can actually reverse this in cells. Shinya Yamanaka actually won the Nobel Prize for discovering 4 factors that can convert an old cell back into what looks like an embryonic stem cell. And later, as scientists were applying things like the epigenetic clock to this data, we found that not only are you changing the cell type, but you're also erasing or essentially reversing all those epigenetic changes that we've used to try and quantify biological age. We always thought aging really happened in one direction, and that the only thing you could really do was just slow the accumulation of this damage. But really what this reprogramming of the epigenome tells us is that this is a lot more modifiable and elastic than we originally knew. So the question becomes, how do you do this in a body? Can you actually program cells from an old epigenetic state back into a younger epigenetic state? What does this actually mean for our physiology and our health? Right now, people are using the epigenetic clock as more of a diagnostic, as opposed to a means to intervene. So people are using it as potentially one indicator of how they're aging overall. It's not a perfect indicator, but it can give people some indication of their health status and potentially their overall risk of developing different diseases of aging. A lot of the changes that cells undergo with aging, including changes to the epigenome, give rise to some diseases like cancer. So the risk of cancer increases exponentially with age, and we think some of this might be due to the types of changes that are measured when we look at the epigenome. The reason why scientists are so excited about the idea of intervening in the aging process, whether it be slowing the aging process or reversing the aging process, is because we actually think that in doing so, we can stop all of the different changes that are giving rise to the diseases that we care about. So rather than going after one disease at a time and having one type of science aimed at trying to cure cancer and another aimed at diabetes, if we actually could reverse or slow aging, we could basically eliminate diseases, or at least postpone diseases across the board. So are we going to solve death, and will the epigenetic clock help us get there? My thinking is that we probably will not solve death, and this actually shouldn't be our goal. What our hope is that this will just postpone disease, but we're not going to, quote, cure aging or cure death overall.
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. One once we understand how our epigenome influences us, we might be able to influence it too.
Doctors and epidemiologists, nutritionists who discovered the kind of foods and lifestyles that lead to a healthy lifestyle, a healthy life, and also longevity, such as the island of Okinawa or the Mediterranean diet. They discovered these things independently from us who are working on genes and enzymes. But what we've discovered is that the molecules in those foods and those types of exercise turn on these longevity genes. There's sirtuins, but there's others. There's one called mTOR that responds to the amino acids and protein that we eat. There's one called AMPK, which responds to how much energy our cells have and how much sugar is in our body. And so in other words, what we've learned so far is that these types of diets and exercise, I wouldn't say inadvertently, but not coincidentally, are turning on these genetic pathways. But what's important is that now that we know that, we can make medicines that are 100 times more powerful than you could ever get from a treadmill. And not only that, you cannot expect an elderly person to go running on a treadmill for 10 minutes a day. What we need is an IV or a pill that we can give them to revive them.
So what do you do for you in your own life, apart from those things that you've discovered that's a handy tip for, in other words, that you've learned that you're trying out for yourself and not just your resveratrol from red wine? I mean, what are you, in terms of your behavior, your lifestyle?
Yeah, well, I've become a little bit of a role model here, not really intentionally. I'm just a scientist trying to do the right thing. But the older I get, I find that the more interested I am in my own research. But so I've just turned 50. My father's 80. We're both on a very similar program. We monitor what we do very carefully with rings like this and blood tests that you can get commercially, at least over here in the US. And we see what works for us and what doesn't. So we're not just blindly experimenting, but we are trying to see what happens in a very safe, controlled way. My father's also a scientist in Sydney. He can judge for himself. So yeah, resveratrol I've been taking for 13 years. What's that?
Go on. Sorry. Yeah, what's that?
Oh, so this is called an Oura ring, O-U-R-A. Kind of. It'll tell me heart rate, movement, sleep patterns. It's very useful for optimizing sleep. That's one of the great things about it. Of course, we've also got these devices now as well. So my point is that if you just pop a pill or do some exercise and you don't measure yourself, you're flying blind. Sometimes I just cut to the chase and I say I wrote down what we do in my family on page 304 of the book that I just put out. But I'm not trying to sell books here. I'm just trying to say if we don't have enough time to get through it, people can go see what we do in a list. But it's resveratrol. I do NMN. I take a bit of NMN, which is an NAD precursor that raises NAD. I'm taking more olive oil since it was just discovered that oleic acid activates one of the main sirtuin pathways, just like resveratrol, so that's good for us. Vitamin D is important. And also I'm taking metformin, which is a drug that is probably the craziest and most dangerous idea that I'll bring up, which is that there are drugs on the market like metformin for diabetes, type 2 diabetes, that have been shown in tens of thousands of people to slow down the rate of age-related diseases. So not just diabetes, but cancer, heart disease, Alzheimer's, and frailty. But you need a doctor's prescription to get that in Australia and the US.